So I think only presenters can record. Do you figure out? Okay, my screen said someone's recording this meeting. Oh, I don't know, but I am now. If you have, if you have the Skype for Business downloaded there, okay. You usually have to have presenter status to be able to record, and not all of you do. It depends on how you check in. But I'm recording it now and can upload it. That's fine. Okay. All right. So, any questions about what we've talked about so far? Okay. Okay. Yeah. Because sometimes certain things we go through pretty fast. You might have been like, "Wow, that was really fast. Holy crap." Okay. Now the mitochondria. This is what some people only remember from sophomore biology is that the mitochondria is the powerhouse of the cell. But it is because it is the place where energy is produced, the ATP, adenosine triphosphate. That is the body's source of energy for all most chemical reactions that occur. So we need to produce a lot of it in order to keep our body running. Um, when you take in food, so here's going to be my attempt to explain this. You don't have to get crazy with the biochemistry of it, but there is a part of it that you need to understand because it helps explain the symptoms that people feel and why prolonged decreased oxygen will negatively impact cellular function. So we as humans require oxygen for effective metabolism. When Here is my little cell. When I take in glucose, glucose should transfer into my cell cytoplasm. How does glucose get into the cell? Trick question. I got some typing. I'm excited. Insulin! Oh my gosh, I'm so happy. Sometimes it's painful for that one. Yes, it does allow that large glucose protein to get through. When that glucose protein, that's not a protein. When the glucose gets inside the cytoplasm of the cell, this is our source of energy. So that glucose inside the cell is cut into three pieces. That lysis requires the use of ATP in order to do that. But that's okay because these both can produce ATP themselves. So this was a negative, we had to use it, but here we have positives. So we end up with a net of 2 ATP without oxygen, just by the glucose coming into the cytosol and being cut. We get 2 pyruvates that we can get ATP from, and our leftover is lactic acid. So we did get energy because we need energy for cell function, and we had a byproduct of lactic acid. Does anyone remember ever feeling a buildup in lactic acid in their own cells? Have you ever exercised? It's been a long time, so I'm wondering what it feels like. Pilates, yeah, that would do it. So it starts to hurt, you know. When, yeah, definitely. When you add oxygen, you can help reverse the lactic the lactic acid process, and get rid of some of that so that that pain goes away. So yes, that buildup of lactic acid is painful, but also when you split the glucose and end up with half and half, you don't get as much ATP as you could if you could have used oxygen. So if I would have gotten to use oxygen, then those two pyruvic acids would actually join with oxygen and become acetyl-CoA, and they'd be able to enter the mitochondria. And they go through this huge electron chain reaction, and the end product, waste product, is carbon dioxide and water, and you get lots of ATP, 36 ATP. So I got lots more energy, and my waste products, instead of being lactic acid, are things that my body is trained to get rid of, carbon dioxide and water. So it's not painful. My body can get rid of those agents easily, and I have a much greater energy production, which will be very important. It'll make more sense about hypoxia can cause cellular injury. So we certainly know that hypoxia would decrease our energy production and can lead to pain for us for sure. And so that's the oxidative phosphorylation process that I just sort of scribbled for you, that the small particles enter the cell cytoplasm and get converted to pyruvate. But if I wanted to get the most bang for my buck, I would be using oxidative phosphorylation so I can get into the mitochondria and actually get large amounts of ATP. The other parts of the cell that we mentioned are the cytoskeleton, the microtubules and filaments that provide structure and movement for the cell. Depending on the type of cell and its job, it might contain more tubules or filaments than others. If the cell has cilia on it, it will definitely have a lot of microtubules. If the cell is meant for movement, like microfilaments are part of muscle contraction, so muscle cells have lots of microfilaments. So there can be a different proportion of those per cell, per function of the cell. The next thing we're talking about, or before we talk about cellular communication and movement across the membrane, is the cell membrane itself, which is semi-permeable lipid bilayer, gives the cell structure. It's sort of like a peanut butter and jelly sandwich there. The two pieces right here and right here stick together, and because of these hydrophilic heads that like the water and hydrophobic tails that don't. So the tails gravitate towards each other, and then the water sides are on the interior and exterior of the cell. So the cell membrane is useful. It's semi-permeable, meaning it'll allow some things through, not everything. We don't want everything to come into the cell or it would destroy the cell. So it's selective. Fluid can move in and out of the cell. Water can move in and out of the cell. Smaller particles will have an easier time moving in and out of the cell, especially if they're not polarized. Larger particles won't just be able to diffuse through. It would have to be permeable. They'd have to be able to go through. And if they're really big, they're going to need help to get through. The cell membrane, it has other embedded or integral proteins in it that are part of channels, that are part of, let's see, there's glycoproteins and sugars that help connect cells together. Cell membrane, here's just written out the lipid bilayer, integral proteins, peripheral proteins that can go through the other side of the membrane. This cell membrane also helps the cells stick together so they don't just go floating away. It helps in cell-to-cell recognition, and it's one of the things that helps the cells not grow out of control. They know they have to stay within a certain space. There might be receptors on the cell membrane for hormones. This is where electrical events occur across the membrane, and as I said, it aids in regulation of cell growth and proliferation. So how will cells communicate with each other? They have four main ways in which they do this: autocrine, paracrine, endocrine, and synaptic signaling. Autocrine signaling means that the cell created a substance or chemical that it releases, and that substance or chemical affects the cell itself. So it's doing something to its own self. Paracrine signaling means it's releasing a chemical and it's affecting nearby cells. So we'll talk a lot about autocrine and paracrine signaling in the next unit when we talk about the immune system and the inflammatory process. There's a lot of communication to the cell itself to increase replication, signaling to nearby cells as part of the immune process, which might actually help in targeting and killing cells that don't belong there. So we'll talk about that kind of communication. We won't talk as much about endocrine signaling until towards the end of the semester when we talk about more of the endocrine system. But in that case, a cell produces a hormone that leaves the cell, is carried off in the bloodstream and goes someplace else to do its action. Synaptic signaling, that's in the middle of the semester that we talk about the way nerves communicate with each other through neurotransmitters. And that's just pictures of those same processes. This next section, the types of communication and the movement of fluid across the membrane and how that occurs is very important coming up for the first exam. There'll be some questions about that. Like I said, there'll be a study guide, but this is definitely important to be sure you understand it. Some of these you might remember. To be a passive process of movement means that no energy is required. To be an active process, then ATP is required, energy is required. So that's the main difference between active and passive. So those first three under passive transport, diffusion, osmosis, and facilitate diffusion, none of those transport mechanisms use ATP. The active one obviously does. Secondary active transport means that molecules are taking a ride on the ATP that's being used. So the diffusion is movement of substances down a concentration gradient. The body always wants to keep concentrations in balance. So in this picture, that middle thing is the semi-permeable membrane. One side has a high concentration of a particular solute, the other side has none. As long as that membrane is permeable to the substance, then it is natural for the body to try to equal out the concentrations. So just the fact that there's an unequal distribution of that particle between the inside and outside of that membrane means that as long as it's permeable, those substances will passively move down their concentration gradient. If this substance that was in unequal concentrations between a membrane is not soluble, then it'll be the water that moves instead in order to equalize the concentration. So in this picture, it's that right side that has more of a concentration of whatever particle that is than the left side. But because if that membrane or that like dotted line that's represented there, if the particle was permeable, then it would just cross over to the other side and equalize the concentration. However, it's not, but water always is. So water moves in order to equalize the concentration. So the biggest difference between diffusion and osmosis in deciding if it's going to be the water or the particle that moves down its concentration gradient is whether or not the membrane is permeable. You can sometimes have what's called facilitated diffusion. So we just talked about simple diffusion, which means that something that's permeable will diffuse across the membrane into an area to equalize concentrations, and that doesn't use energy. However, you could use a transporter too, which will make this happen faster, but it doesn't necessarily mean that energy was used. So this example right here, the channel protein is open, allowing a negative ion through down a concentration gradient. It's hard to get charged ions through without assistance because they might be too polarized. So the channel protein didn't need anything to open it, it was just open. So they got to passively diffuse across with some assistance. And we can see how diffusion and facilitated diffusion work when we talk about the action potential coming up. The other way, which is also important when we talk about the action potential, is primary active transport, meaning that if we want particles or molecules, ions, whatever, to move against their concentration gradient, to go in the opposite direction, to go from a low to high concentration, then you need energy to do that, and you will need ATP. And so the example is the sodium-potassium ATPase pump, which we'll talk about when we talk about the electrical potential. But that will allow exchange of potassium and sodium. You'll get the exchange of 2 potassium and 2 sodium ions against their concentration gradients because you used energy. As I mentioned before, secondary active transport means you are using the energy to transport or facilitate the movement of a substance, but you're using the energy from a different pump. So symports or antiports. I could, because one substance is diffusing through, someone could ride along, or they could go in opposite directions. So here is when we're going to talk about the membrane potential. So how much do you guys remember about the action potential or electrical potential in the cell? Oh, Dawn is going to wow us. I'm excited. Yes, the cell membrane's resting potential is negative. That is true. No points removed for spelling, Dawn. I'm amusing myself. Good deal. It's Karis, right? I keep wanting to say Charisse, and I'm telling myself to stop it.
I should just talk. It's Karis.
Oh, God! Okay. Damn it, I'm writing it down.
So the cell pumps sodium in out and potassium in, and it creates a negative charge. And then when the gates are released due to some kind of stimulus, sodium and potassium both reach equilibrium, and then the whole system starts again.
Yeah, that's pretty good. So a lot of cells use the action potential in order to send signals. This is especially something that occurs in nerve cells and muscle cells. You are correct when you said that the resting potential is generally kept negative, meaning if I put a voltmeter into this plasma membrane and tried to measure the electricity, my count would be about negative 70 millivolts, negative 60 millivolts there, if I put a little voltmeter there. And the reason it's kept that way is because of its usual balance of ions. And Karis, because I wrote it down, mentioned that we do tend to keep potassium inside the cell and sodium outside the cell. Potassium is the major intracellular ion and sodium is the major extracellular ion. Their negative ion in the cell is proteins, which we just talked about. The cell is made of lots and lots of proteins, a huge part of it. Out here, it's chloride. So between this balance, we have more sodium inside, more sodium outside, more potassium inside. Sodium kind of wants to move down its concentration gradient because there is one. And it's not that it's not able to diffuse, just not very fast. So once a certain amount of sodium gets in here and we start to get more positive, that's like flipping the switch. So it's and these channels open. And just like Kara said, these channels could open because of a stimulus as well from a change in um, the nearby cell activity, from a paracrine reaction or a synaptic reaction. And when that channel opens, sodium is allowed to pour into the cell. So very quickly the action potential occurs, or the cell flips from being very negative to very positive. And then when that occurs, those channels close. Because now we have this concentration gradient between potassium in the inside and the outside, potassium will start to slowly efflux or come out of the cell. But then what helps us really return back to resting potential like we want to is the sodium potassium ATPase pump, because it's going to force sodium outside the cell and potassium inside the cell against its concentration gradient so that we can get back to resting potential. So we had passive diffusion, facilitated diffusion, and active transport all working in order to keep a resting potential, generate the action potential, and return to resting potential. I do remember there's a hyperpolarization. That's true. So you were right. As a certain, there is a time called the refractory period when you're too positive to accept a new impulse, and until that you start to get closer to the resting potential, will the cell be able to accept a new impulse? That is very true. Any questions about that? Because you can see later on in the lecture, we talk a little bit about sodium and potassium concentrations. So obviously it's going to affect cellular function. And you see in that picture that I drew, that for whatever that's worth, that this part, this reestablishment of resting potential, will require energy because active transport requires ATP. So keep in mind that if there's a problem with the production of ATP, there will be a problem returning cells to their resting potential, and it'll affect sodium and water balance. So that is describing what I just said. And that's a cute picture that's better than mine of the diffusion and through that channel that is open, so it's facilitated diffusion and active transport. Any questions before we start to talk a little bit about types of tissue? Oops. So the reason, some of the more important things from that first part of the lecture that we sort of whipped through was the importance of DNA and no mutation in order to support continued correct cellular functioning. And if there's mutation, it's best if the body can find it through its natural surveillance and program that cell to die, which we'll talk about too. We talked about types. We talked about the cell membrane. We talked about lysosomes, which we'll talk a lot about in the immune system functioning. We talked about the cell membrane and how that keeps the cell from just exploding. It helps give some control to the cell about what can go in and out. We talked about cellular communication, autocrine, paracrine, endocrine, and nervous, synaptic transmission. And then we talked about diffusion, facilitated diffusion, osmosis as the passive mechanisms of transport, and active transport using energy, and then how that relates to the action potential. So hopefully at least that stuff made sense. Now, this might be getting a little closer to things that you can actually see, which is sometimes a little helpful. We're going to get towards the types of tissue and how that's organized within the body. So here's the list of the types of tissue. Um, I'm still sharing. Okay. Epithelial, connective, muscle, nerve, and then the extracellular tissue components. Epithelial tissue is the most abundant type of tissue. It covers and lines all body surfaces. Whatever is in the free surface determines its function. Epithelial tissue lays close together to keep a barrier, and it adheres together to try to also control growth of the cells. So there's the next picture or picture coming up is the types of epithelial cells, but in general, they are very close together. They are stuck together with adhesion molecules that keep them anchored on a basement membrane, and they'll be fed by a capillary or blood source nearby. Classifications of epithelial tissue depends on the amount of layers and its shape. So epithelial tissue could be simple, meaning it's just one layer, stratified, meaning several layers, pseudostratified, meaning it kind of looks like it's layered, but it really isn't. Classification is shape, squamous, cuboidal, or columnar. So this is an example. So you can see simple squamous, stratified squamous, so that's layers of squamous cell. Simple cuboidal, stratified cuboidal, so those are kind of what I drew. They're just the cubes. Columnar and transitional zones. Wherever there is a transition of epithelial tissue where it might have started as squamous and changes to columnar or cuboidal is an area sometimes that is sensitive to cellular mutation or changes, excuse me, leading to mutations that might actually lead to cancer. So one of those transition zones is present inside the cervical os, where squamous epithelial cells that line the vagina and part of the cervix transition to columnar cells. So when you're getting a pap smear, those of you that will end up doing that in your track classes, you want to get that little spongy area into where the transition zone is, because that's the cells that are at risk for cellular transformation to cancer. So even you, if you get your own, now that we have my charts where you can actually see the pathology of the things that you have. If you ever got a pap smear that said normal squamous epithelial cells, then you need it repeated because it should say transition zone or squamous columnar transition zone present. And if that's negative, then you're good, because sometimes people don't do their pap smears correctly. So it is good to know which type of cells are expected in certain areas. Connective tissue connects and binds and supports tissue, so it's fatty tissue, it's connective fibrous tissue, ligaments, collagen. It contains the bone and blood cells. That's all examples of connective tissue. Muscle tissue, we will talk about more when we are in the nervous system information, because we talk a lot about the nerve and muscle junction. So we'll get into that more as we get into class. But there's the filaments lined up and the interface between actin and myosin for muscle contraction. And then nerve tissue is sensory and motor function interacting with our environment. And again, we'll talk about that more when we get to that portion of the class. And extracellular tissue components. So this is the tissue that is surrounding those epithelial cells. So we have this looks like, I guess it's probably columnar epithelial cells. This yellow line this has a basal lamina, which is a basement membrane. And underneath there is collagen and fibrin supportive tissues. There's our capillary giving blood to this area. There's mast cells that always live by capillaries, fibroblasts that are there ready in case we need to perform wound healing, macrophages that are there just as surveillance cells just in case, glycoproteins that provide structure as well. Those are all together with the tissue. And this composition, there's another example here. It's the fibrous adhesive proteins that are found in the basement membrane and the protein glycogens. That's the composition of our tissue. So we got through cell and tissue types. And the next part, which we can start before our next break, is actually starting to get to the good stuff, the good pathology stuff, is how will these cells respond to stress? And some stress that cells respond to is related to normal stresses, normal growth and development. It's the things that we expect. That's physiologic stress response or expected stress response. Some that we see is not expected and is an example of what can occur because of a pathophysiological problem. So that is our next thing to discuss, which is cellular injury and response to stress. So cellular response to stress takes several forms. Some of these can occur, this atrophy, hypertrophy, hyperplasia. Some of these can occur as a normal part of growth and development. Some of these, metaplasia, dysplasia, intracellular accumulations and calcifications, don't really have a physiologic reason, but can occur related to types of stress the body encounters. So this is a visual representation of what those things mean, but we're going to talk about them all in more depth. So atrophy is a decrease in cell size. It will happen because of disuse, loss of innervation or stimulation, or loss of blood flow. So there's all kinds of reasons that we can think of that someone might get atrophy of tissue that is pathophysiologic. Someone has a severed spine, they would lose nerve innervation to muscles, which will atrophy. Someone has loss of blood flow to an area with lack of oxygen, there'll be necrosis and atrophy. There is situations when atrophy occurs as a normal part of development, and it's when there are structures that we needed at a certain time in our life that we don't need anymore. So in the first year of life, while we're developing our immune clones, when we're developing our own population of immunoglobulins and B cells and T cells, we use the thymus, which is a gland that's substernal, and that is a larger gland, an active gland in the first year of life. After that, because it's not needed anymore, those cells that were created in there migrate to the areas they're supposed to be in, like the spleen and the bone marrow, and we don't need the thymus, and so it atrophies, and for adults it's not detectable any longer. Person who is premenopausal, who goes through active cycles, menstrual cycles, has a normal sized ovary, and then after menopause, when there's no longer estrogen stimulus, the ovaries atrophy and are very tiny and sometimes unable to detect on a bimanual exam. That would be physiologic atrophy. Here's an example. The person was in a cast. They weren't using that leg, so disuse led to atrophy of the muscles. Stuff like that would be easy to get back. But those are all examples of cells getting small. Hypertrophy is an increase in cell size, and that's related to an increase in work demand or stimulation. So sometimes we do that on purpose. If you go to exercise and lift weights, the thought is that you're going to increase muscle mass, increase size of muscle because you increased tension on that muscle and made it work more. That was the point. However, there are times when hypertrophy related to increased work is a disease state, such as left ventricular hypertrophy. So this is a cross section of the left ventricle. When we talk about the cardiovascular system and we talk about hypertension, hypertension is the primary reason why adults develop heart failure. This is the first part of heart failure, which is related to hypertension, is hypertrophy. If the heart is constantly working against resistance, that muscle is stimulated and will get larger. Hyperplasia is an increase in the number of cells. It could be physiologic. We're allowing for organ regeneration in areas where that's possible. We're allowing for, during the wound healing process, we're trying to epithelialize and close up a wound. It could be that it's in time of puberty and there's increased growth and hormone release in order to increase the number of cells when you're developing secondary sex characteristics. So that can be normal. There could be excessive stimulation that's pathologic. So the example here is the pathologic estrogen stimulation of the endometrium post-menopause, which can occur and be the starting point or issue or lead eventually to endometrial cancer. So the reason, one of the biggest risk factors for endometrial cancer is obesity, because fat is metabolized into estrogen-like stuff, and increase in circulating estrogen can increase the endometrial layer, could stimulate hyperplasia of the endometrial layer and lead to postmenopausal bleeding from that excessive stimulation. Metaplasia is a change in cell type. So, and this is really not something that occurs commonly as a physiologic response to normal growth and development. This is a response to chronic irritation and inflammation. It doesn't have to be irreversible, but it's not done as normal growth and development. So the one of the best examples, a couple examples of that is you smoke, that's chronic irritation and inflammation to your airways, and so your airways change from squamous epithelial cells with cilia and mucus production to columnar epithelial cells without cilia and mucus production, which puts the smoker at increased risk of bronchitis. The example I have here is the esophagus is meant to be squamous epithelial tissue, and there's supposed to be an intact lower esophageal sphincter muscle around the esophagus leading into the stomach. The stomach's secretions are acidic because of the presence of the hydrochloric acid, and that hydrochloric acid is not meant to touch the esophagus because it's not meant to handle that acid. But if someone has a weak lower esophageal sphincter, and acid irritates and burns the lower esophagus, you can have a change from squamous epithelial cell to columnar epithelial cell, and that is essentially what Barrett's esophagus is that there's metaplastic change to the esophagus. Dysplasia is an overgrowth of cells, but it's disordered overgrowth. So each cell looks slightly different. They vary in size, shape, and organization. It could be adaptive briefly to excess stimulation, but mostly it is thought to be the first step potentially to cancer transition. This is sometimes someone will have a mammogram with some abnormal calcifications, and they'll see atypical ductal hyperplasia or dysplasia, which suggests that woman might have an increased tendency to have breast cancer. Patient has a pap smear and it shows cervical inner epithelial dysplasia. There's some disordered growth of cervical cells, which suggests, if that was allowed to continue, a potential for cervical cancer. The last two we'll talk about before we take another break is cellular accumulations. So if there's an excessive amount of a substance, could be fat from fatty liver disease, extra bilirubin, extra calcium, extra other metabolic accumulations, lead, stuff like that, that one of the ways the body can deal with extra substances is to hide it into cells itself, causing accumulations of those substances, and that can be damaging to a particular cell. So this is showing intracellular accumulation of fat in healthy liver cells. So the liver cells are the purplish stuff, and the speckled yellow stuff is fat. And over time, if that continues, that could lead to inflammation of the liver and potential cirrhosis. Lastly, before our next break, we'll talk about calcifications. So calcium tends to lay down or accumulate in areas where there's been tissue injury or tissue death, because when a cell dies, calcium is released into the intracellular space, and depending on how that cell that died is dealt with by the body, there can be some calcification. Sometimes the calcification is related to excessive serum calcium levels, so it's a way to deal with excessive calcium from types of cancer. So that is another type of adaptation. So we're going to stop here. Does anyone have any questions about what we've talked about so far? Why are because that's a very good question. Why is calcification so common within the heart? So you are seeing, usually depends on what image you're looking at, but it's very common to see calcifications within vessels, and sometimes within the muscle itself, because it's showing you a change in the quality of the medial layer, the middle layer of the vessels, because they're remodeling and changing. Calcium will be pulled into that damaged tissue. Any vessel that has atherosclerosis will have calcium get pulled into that process. So that's the whole. I don't know if you've ever heard of people getting calcium scans. Like, that's the whole reason to do that, is to see if you have blood vessels or tissue that's getting calcified, because that suggests atherosclerosis, which does increase your risk for cardiac disease. So that's what they're looking for. So you're really seeing evidence of hardened vessels by that. Thank you. You're welcome. So at 7:45, we will come back and talk about the types of cellular injury. Some of it is reversible, some of it is irreversible, and if it's irreversible, it will lead to cell death. And when we get through that, we'll be able to do one of our practice case study. So it's 7:35-ish. We'll come back at 7:45. So I'm going to drop out here again. When it occurs, the structure of this the structure that the cell was in remains intact. It doesn't just fall apart. So in the case of ischemic injury to the heart or infarction to the heart, usually the heart doesn't just blow out. There's a piece or a section of the heart that is fibrotic and is not like the section that has maintained its blood supply, but it still has a shape. Instead of liquefactive necrosis, which is liquid-filled or pus-filled. Um, Caseous necrosis is a combination of both. Fat necrosis is related to the breakdown of fat because of lipase or enzymes. Gangrenous necrosis, or saying something looks gangrenous, is a clinical term. It just means that the tissue that you can see looks dead, but it doesn't tell you which types this is. You would have to explain further, or it depends on what the cause is. Whereas programmed cell death or apoptosis is a non-inflammatory process. It doesn't require the activation of the inflammatory process that we'll talk about next class. It can occur because of genetic damage, like we just talked about with radiation damage, or because with replication there was an improper replication of DNA when we were trying to divide to create a new cell. It could be that a cell is worn out. Over time, the cell is replaced and the old cell is removed. And apoptosis is a much more controlled process in which enzymes are activated inside the cell that break down the inside of the cell and create little membrane encased fragments. So the cellular membrane stays intact. It's these fragments that occur, and those fragments can be cleaned up by surveying macrophages. So the area stays clean. Whereas so here's our different types of cell injury. Increased intracellular calcium, we have hypoxia, ischemia, or we have free radical formation. We end up either with the influx of water, we end up with a switch to anaerobic metabolism, or other effects inside the cell. Any questions about that? It's going to come up a bunch of times, over and over, in fact, because many of the processes that occur with different cellular injuries, we have to transfer cellular injuries in general to specific cell types. So this is an example, this is a very simple example of a case study potentially that could look at a type of cellular injury. So we have this 60-year-old man with chest pain and shortness of breath. The pain is in the central chest, described as swelling. He's found to have an MI. So if I was looking at this first part of the case study, we know that case studies were always looking for the positive or abnormal symptoms. We have our chest pain and shortness of breath. And we found out that it's related to ischemia. So if I was going to write this up for my case study, I would have this crushing chest pain and shortness of breath. These are both related to this myocardial infarction, which we know is related to ischemia. There's decreased blood flow to a section of the heart. So if I know that there's a decrease in blood flow to a section of the heart, that means there's less oxygen to some section. So why does it hurt? It is the lactic acid buildup specifically. So this is cell death is occurring. So it is, but to be specific about the pain, we have to describe the pathologic process that's occurring. We know that, I say, high five virtually. Yes, it's the lactic acid buildup. So if this was my assignment, then I would say that, you don't have to get into as much because we haven't talked about that yet, about what causes a myocardial infarction. But if somebody is having decreased blood flow, a decrease in oxygen to cardiac cells means there is a decrease in ATP production. We're going to be in anaerobic metabolism instead of aerobic metabolism. And that means there's less ATP and more lactic acid. And that means the lactic acid is what causes the pain. So if I was going to answer that question, I would have, although I know you're right, the cell is dying, that's why it's occurring, I have to say why I feel the pain, and that's because of the switch to anaerobic metabolism that leads to increase in lactic acid. So they're short of breath could be, what do you think about that? We haven't really talked about cardiac function, but most of you are nurses. Increase workload, increase the release of calcium, increase O2 demands from dying cells. Yeah, so it's actually a combination of a lot of those things at the same time. So if anytime that a person increases their breathing, besides thinking about anxiety or pain or issues like that, it's an adaptive compensatory action of the body to increase the respiratory rate in order to increase oxygen. So that could be from a lot of those reasons that you guys stated. We're trying to breathe off acidosis. We're trying to get rid of acid. We're trying to, if the patient's heart is not working correctly, as Dawn stated, that this ischemia is going to cause irregular muscle function, which is true, and an increased workload on the heart, then the patient's heart isn't going to pump the same efficient amount of blood into the system, which means the body is feeling oxygen deprived, and that will increase oxygen demand and will cause the person to increase their respiratory rate. So that's kind of how you have to think about it. How can I put this puzzle together? Now, 6 months later, the patient has a cardiac nuclear med scan, and there's an area of defect, a cold spot, which usually means fibrosis or non-functioning muscle in the left ventricle. So what does that mean? What occurred? It's exciting because I see the little pencil. Yes. Cell death has occurred, and we know that it was a necrotic death. And I agree with Karis that we can say it's coagulation necrosis because ischemic damage usually affects the calcium that we were talking about, and that causes denaturation of proteins of the cell, but they maintain their shape. It's not liquefied. We didn't have exudate production. We didn't lose its structure. So there's still structure there. It just isn't functioning correctly because that area is dead. So just from the information that we've done so far, you were able to explain some of the symptoms of a myocardial infarction and some of the findings you would see on a test. And that's an exam that the patient had. And that's what the case studies are supposed to do. They're supposed to draw the connection between the patient and the process. So it'll be similar to this. You got to find the things that are wrong and explain what's wrong. Any questions about that? Looks like you guys are understanding it, which is great. Because it's going to come up a bunch of times. So there is some information that is on our thing here about neoplasm. I just put this in here. I mean, neoplasm will be our big focus, or cancer will be our big focus when we have our cancer presentations. I don't necessarily want to spend a big time on this. It's just to help you when you're trying to understand some of your patient's symptoms and how cancer works. So I just want to briefly talk about part of it. And again, like I said, you will get more detailed patient information via email. Usually when we meet in person, I make you pick out of a little cup, but we don't get to do that. So neoplasm just quickly is, neoplasm is abnormal cell growth or cell proliferation. Cellular proliferation is supposed to be balanced, that we replace cells that are worn out. We don't grow more cells than we need. And there are checkpoints or ways that the body tries to ensure that we don't have more cells than we need. When a cell is induced to divide, there are stop points or checkpoints here looking for cellular errors, especially DNA errors, so that can either be fixed or the cell can induce apoptosis, which is that programmed cell death. There are cyclins and cyclin-dependent enzymes that help increase the cell cycle speed and increase replication when needed. There is supposed to be a regulator, this cyclin-dependent inhibitor. I bring this up because there are some cancers that manipulate the cell cycle and increase cell cycle even if it should be controlled or the cell should be programmed to die. There are inductions of growth. So some cancers, sometimes the body will release growth factors normally in order to induce cellular proliferation for things that we need, like wound healing when we talk about it. But sometimes there will be proto-oncogenes or proteins that are pro-cancer that induce cellular proliferation. And there's supposed to be inhibition when we don't need cells, when there's damage, or when there's hypoxia. This is the specialized protein, P53, that is the surveillance protein, one of the major ones in the cell that watches for mistakes in the DNA. And people can have genetic abnormalities in the P53 protein, and so they don't have scheduled apoptosis like they're supposed to. That's one of the genetic cancer syndromes that you might come across depending on which cancer you have. So cells proliferate when needed. They increase specialization with each division. They start out as a stem cell, and as they divide, they sort of commit to a cell line and then become the specialized cell. So a stem cell, and we'll talk more about stem cells in the setting of the bone marrow, not next class, but the class after, that has the ability to renew itself and has the ability to keep a copy of itself. So here's the stem cell. It can divide and keep a copy of itself, so we never lose the ability to get to this particular cell line. And each time we divide, we become more differentiated and more specialized. So neoplasm, oh, is someone talking? Does someone have a question? I was all excited. You faked me out. Okay. So neoplasm is an overgrowth of cells, and it can be benign, meaning that the cells are growing and proliferating more than they should, but they maintain differentiation. They're not that abnormal looking. They're close to normal cells, and they don't try excuse me, to leave or invade other structures, which is what happens with malignancy. There's less differentiation. They don't look as much like they did before, and they can invade and spread. So what causes a normal cell to become a cancer cell is something has to initiate or expose that cell to make them susceptible to transformation. So it could be exposure from UV radiation, which we just talked, which could be a cell cancer initiator. It could be HPV infection, human papillomavirus, which can cause cancer initiation in normal cells in the head and neck or the cervix. And that leads to unregulated and accelerated growth in cells that eventually can require malignant changes. Cancer has several things in common with each other in that they have nothing that stops their growth. They can continue to proliferate even without stimulation. That most cells have these adhesive molecules that make them stay together, and but cancer cells lose those adhesion molecules and are able to leave spaces they're not supposed to easier, which is loss of cell anchorage, which means normally if a cell falls off its anchor to its neighbors, it would be killed, programmed cell death, but a cancer cell won't. It'll just be able to travel. It'll change its function and change the way it communicates with other cells. It changes how the chromosomes shorten. Telomeres are at the end of chromosomes and prevent the cell from dividing too long. Each time a cell replicates, a piece of the telomere cap falls off. And once the telomere is gone, then this should program the cell to die and not allow for continued replication. But some cancers never lose their telomeres, and so they can just divide forever. There can be other hormone changes, cytoskeleton changes, and what can happen then is tumor cells can create enzymes, so it's easier for them to break through the basement membranes and into blood vessels, and easier to travel and metastasize other places. Do I have an example of cell anchorage falling off? It's one of the ways that cancer can metabolize. So cancer cells have this ability to leave where they are, and they have enzymes that allow them to move other places. So here's actually a detached cell. This is a tumor that started. In order for a tumor to continue to grow, it must create its own blood supply. So it releases vessel growth factors in order to feed itself. And as it gets really large, it's easier for the cell to detach itself. Now, if this here, what's happening, oops, wow, that was cool. What's happening here where this little cell is falling off, in a normal situation, that cell would die. But in a cancer situation, it doesn't. It would just float and land wherever and could potentially start growing someplace else. It could use enzymes to break through into lymph channels, or it could just squeeze through lymph channels or blood vessels and travel someplace else and grow someplace else. So because they can lose anchorage means that they can end up in the bloodstream or the lymph channels. So I'm not sure. Hopefully that answered your question. Any other questions about that? It's a pretty fast rundown, but just a little bit of information about what malignancy is that might be useful to you when you're starting to work on your presentation. And I thought I would just bring that up since we were talking about cells. The next part that we're going to talk about is fluid balance. I know, right? Isn't that impressive? Now you're all oncologists. There you go. That's hilarious. It's true, but it's really the important thing to know is that cancer figures out a way to not get killed and to keep growing because that's its thing. I don't know if anyone's ever read the book or seen the movie The Immortal Life of Henrietta Lacks. So excellent. Yeah, so there are still HeLa cells that are dividing as we speak. They were from her very poorly differentiated cervical cancer and have been used to create targeted therapies to understand cancer growth, been used in tons and tons of labs everywhere, all over the world. Of course, like most things in the US, has a background story of racism and um, use of people without telling them, like her cells, because of how poorly differentiated they were and how much they divided, were taken, but the family was never aware of that. I mean, the family should be billionaires, millionaire, trillionaires, over 1,000 times over. So many people have used those cells. Um, anyway, it's a really good book. I know you have time to do lots of reading. I'll show you. There's a really cool video of how HeLa cells still divide, if you feel like watching it, just so that you can spend a couple more minutes on cancer. Um, but anyway, the next section we're going to talk about is fluid balance, and two of the main pathologic disorders that go with that, or have the potential to be a problem for people, is edema, which is fluid leaving and going into interstitial space, and dehydration, which is of course fluid leaving the compartment it's supposed to be in. So these are pictures of very different types of edema. This is a graph showing you the breakdown of total body fluid, which is fairly similar between men and women, that most of our body is made of fluid. About 55 to 60% is made of fluid. And of that fluid, it's divided into two compartments. The largest compartment is intracellular, inside the cells. The other compartment, the smaller compartment, is the extracellular fluid. And extracellular fluid is further divided into interstitial fluid and plasma. So when we're thinking about fluid and fluid compartments, we have to remember again how water moves, and water is easily moves from place to place, and it's drawn by osmotic forces. In order to equalize concentration between fluid compartments, water can move fairly easily. And we can talk about how water can move between the intracellular space and the extracellular space. Kind of like we did, it was a pathological instance, but the cells with the ATP, depleted ATP that couldn't run the sodium potassium ATPase pump, ended up with excess sodium, which was the draw for fluid to leave the extracellular space and go into the intracellular space. But we can also think about water movement between the extracellular spaces, between the inside of the vessel and the interstitial space. And that is the process of filtration, which is extremely important. It's the water movement between extracellular fluid compartments, specifically between the vessel, the plasma, and the interstitial fluid. So it's that first picture that we looked at. It's this part. Oops. This part right here. Oh, nothing wants to work for me. Right there. Them moving back and forth here. And it's in general talked about as net filtration. So we'll bring up Starling again when we're talking about the cardiac unit, but Starling said, or Starling's hypothesis is that net filtration, or the movement of water from the vessel to the interstitial space, is equal to the forces favoring filtration minus the forces opposing filtration. So don't get stressed out about that. It's not as hard as that sounds. So if, oops, here's our blood vessel, and here is our blood vessels are lined with special cells called endothelial cells, very different than epithelial cells. These are specialized cells that line vessels. So here's our blood vessel. This is where our plasma is. And this is the extracellular fluid space, the interstitial fluid, the fluid between cells. Filtration means, just like if you ran water through any kind of sieve, you're sifting through. Filtration means we're going to have fluid move out of the blood vessel between these endothelial cells, kind of filtering fluid through this vessel membrane into this other space. So I always think of it as like a sieve. It's a filter. Or if like you're filtering your water with a Brita filter, I'm only going to let some of it through. And the forces that favor filtration is capillary hydrostatic pressure and interstitial oncotic pressure. So let's break that down. That means that there's a lot of water. There's an increase in water. So that means there's a concentration difference between water inside the vessel. There's more water inside the vessel than there is outside the vessel. So that would favor filtration because I have more water here and less water here. That favors osmosis. That favors filtration. If there's a lot of protein in here, in the outside of the vessel, in the interstitial space, then we increase the concentration. It could be, the body wants to move down concentration gradients, but it's not, so there's less protein in here than there is in outside of the vessel. But albumin is a very large molecule. It can't just go through and filter through the vessel unless there's a problem. This protein is stuck here, so the protein can't move down its concentration gradient. And when a membrane is not permeable to the solute or substance, then the water will move instead. So that's another draw for water to filter out of the blood vessel and go into the interstitial space. So those are forces that favor filtration. Lots of water in the vessel and not a lot of protein or more protein outside. Forces that oppose filtration are the opposite. That there is a lot of protein or an increased protein in the blood vessel and there's not as much water. So there's more water out here in the interstitium, then that's going to stop water from leaving the vessel. It'll oppose filtration. So if these forces that favor filtration are greater than the forces that oppose filtration, then you will have fluid leave the vessel into the tissue, which is potentially the cause of edema, depending on what has caused those things. Does that make sense? Can anyone say it back? Candy's on board. Yup, awesome. So the forces of filtration are important, high to low, yep, because they can be normal. We can increase the force of filtration, like when we talk about the inflammatory process next class. We can cause fluid to leave the vessel by manipulating that. That's funny. We can understand why some disease processes, which we'll talk about, could favor movement of fluid from the vessel. Can you think of any disease process that would cause edema, that would make fluid filter from the vessel? Yes, and that is part of the inflammatory process. And we will talk about that a lot more next class. Heart failure and liver failure and kidney failure. So those are very good causes. Heart failure oftentimes means that fluid is backed up in the system. Instead of moving forward into the arterial system, we have too much fluid in the venous system. And when there's increase in hydrostatic pressure or standing water, that is a force that favors filtration. Increase water, high to low, just like our friend said, Miss Manka. That means we're going to have fluid leave the vessel space and go into the interstitial space, which we will appreciate as edema. If the person has kidney failure, like Gina said, it's also the same. If the person is still taking in a lot of fluid, but they're not filtering that fluid, then the fluid is stuck in the system. That's an increase in hydrostatic pressure. His first name is pronounced like Erica? Okay. What did I say, Erika? I don't know. Who knows what I am thinking half the time. Um, and then, so that's another increase in hydrostatic pressure. The liver disease ha, can lead to increase in filtration because the liver is responsible for the production of albumin. So yes, so if I lose albumin, then I have decreased oncotic pressure inside the vessel, especially in comparison to the protein or oncotic pressure in the interstitium, which will lead to edema. So here's a tougher one. Besides the albumin, why would cirrhosis lead to ascites? Bum, bum, bum. Yeah, so here I'll draw it. It's part of it because there's too much pressure in the liver itself. So one of the ways to do that is to do something to draw off the pressure. But there is, okay, so here I am. Here's my cirrhotic liver. My portal circulation from the small intestine and my inferior vena cava is supposed to run through the liver up into the right side of the heart. So if this is now a rock because it's fibrotic, instead of the spongy vascular tissue it's supposed to be, it's going to be harder to get the fluid through that liver into the right side of the heart, which means an increase in hydrostatic pressure of the inferior vena cava, and that means fluid's going to filter into the abdomen. So you get ascites. Why does a DVT in the great saphenous vein in the leg lead to edema, unilateral edema? Miss Erica. I wrote that down, Erica. Yeah, it's a backup on the side of the DVT. It's kind of the same. So we are going to cause a backflow of fluid, and that means an increase in hydrostatic pressure. And that means there will be an increase in filtration. So we talk here a lot about reasons for forces favoring filtration, which would be something that causes an obstruction of the vein, like cirrhosis or a clot, or external compression from a tumor. Sometimes ovarian cancer will present as lower extremity edema, especially in the right, because there's external compression of the inferior vena cava, and so that could be a possible presentation. Decreased plasma oncotic pressure, we talked about, that has to do with the decreased production of albumin. We will talk a lot about the response to infection, inflammation, including sepsis, but also even locally in a certain area, because increase in capillary permeability is going to increase filtration. And lymphatic obstruction is another reason why people get edema, which is slightly different. It's not because the forces of filtration are off. They could be normal, but if fluid leaves the vessel, there's supposed to be lymph channels in this interstitial space that help to filter excess fluid out, return it to the venous circulation, and then particles that don't belong there, foreign substances, etc., are filtered through the actual lymph nodes. If these are removed or blocked because of cancer or surgery or something, then fluid that comes off has nowhere to go. And that's the other reason for edema, is lymphatic obstruction. So, how are we doing? Is everybody having fun? I hope. So, whee! I like that. So we're going to take our last break. We're getting through. It is more fun than the book. I am so glad you said that. If it was the other way, I would feel really bad. Um, it is now 10:35 or 10:35. Did you get a heart attack? It's 8:35. We will come back at 8:45 and finish as much. We'll probably finish what we need to finish today, which is great. So I will see you in 10 minutes. Um, We will go back to the, oh, I just accidentally opened my Pandora. That's hilarious. Um, go back to sharing my screen here. So that we can look at dehydration. So dehydration, now we are looking at the balance of water, and when some of the water is missing from a certain compartment, or there's not a balance between the amount of sodium or the amount of water. So we know that, especially those of us who have worked as nurses, that the body's sodium percentage is 0.9. 0.9 normal saline is isotonic for our own blood plasma. And if we lose water without a change in concentration, so there's no change in osmolarity between fluid spaces, or between the extracellular fluid spaces, or between the extracellular and intracellular fluid spaces, then we lose water, but there won't be a shift of water in and out of fluid spaces because there isn't a change in concentration. So isotonic water loss is common with sweating or drainage or bleeding, but especially in the beginning. Over time, you're going to start to lose sodium. There's going to be a change in concentration, but in the beginning especially, it's an isotonic fluid loss. So if I lose fluid without a shift of fluid, what symptoms do I have? Gina's typing, so is Amanda. Hypotension, correct. So we are losing volume. We will lose fluid. Thirst is also correct. Loss of volume is a natural trigger to thirst. Tachycardia, dizziness. So the increase in heart rate is compensation for the decrease in fluid volume to try to maintain blood pressure. Dizziness, especially when standing, because the person could become orthostatic. The dizziness is loss of flow to the brain. So those are the symptoms we are going to see with isotonic fluid loss. Pure water loss means we're losing water and there's an increase in sodium. So this can occur if isotonic water loss occurs over time, like you're going to end up losing more water, and so the sodium will increase. Especially with big wound injuries or burns. It's very easy to lose pure water. You can have this occur if you are profusely sweating or you have a fever where the respiratory rate increases. You'll lose pure water from the lungs. Um, Diarrhea, especially in infants, sometimes they call it, especially in developing countries, running belly, where they have such horrible diarrhea that they lose tons and tons of water, that they end up with a hypertonic fluid loss, meaning that if the normal osmolarity, or when we have isotonic fluid compartments, is around 280 milliosmoles. So if I am profusely sweating, have really bad fever, and I'm losing a lot of pure water, I have a big burn, I am going to lose a lot of water. And so the concentration inside the vessel will increase because there's loss of water. So it might be 310 milliosmoles here in the vessel, but still maybe 280 in the interstitium. So where will the fluid go in this scenario? Um, well, remember this increase is in concentration of particles like sodium. So this is increased sodium in the vessel. Right, so it depends on where I'm asking. So if it's between this extracellular space, the interstitial area, and this extracellular space, the vessel, where will the fluid move? Into the vessel or out of the vessel? It's the interstitium into the vessel because we're going to follow the concentration gradient. Because this concentration is high, because of there's more protein in relation to the outside, because the water is gone, the water would leave the interstitium and go into the vessel. So I like to bring this up because who can tell me what turgor is? Skin tenting, right? Right. So that means when a person, especially if I'm not dehydrated and I lightly pinch, some skin, it should fall back instantly into its position. If a person is dehydrated and I pinch their skin, it'll stay up because there's less fluid in there. So that's the reason why dehydration can cause skin tenting. But like Karis is saying, which is true, if I'm solely looking inside the vessel because there are cells in the vessel, there are red blood cells in the vessel. If this vessel is very concentrated in relationship to this cell, the fluid will leave the cell and go into the vessel, which I think is what, if I remember correctly, what Kara said, out of the cell into the vessel. Yes, so it depends on which way you're asking. That is all correct. So hypertonic loss will lead to intracellular fluid dehydration. And so that can mean that for certain areas in the body, that can mean, like we talked about with the change in the elasticity of the skin, but it could be a change in muscle function, and it can be a change in the function of the brain. If the brain cells shrink, we will end up with confusion and seizures over time. So you can't, over time, you would have to, that person would need a lot of fluid. Sorry, hypotonic loss is usually a pure sodium deficit. So it's not necessarily that that the person is dehydrated. It's just that the fluid has moved compartments. And when there's say the serum in this vessel, the serum sodium is low, less than 135, so the concentration of solutes in the vessel is low. So this is low. Which way will the fluid move? Into the cell or out of the cell? Mm-hmm. 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 It's going to go into the cell. Is that what you guys said? I'm sorry, was it? From the vessel into the cells. Yep. It's going to move this way. So cells get larger. That's still bad. So that means that in this extracellular space, there's less water. But it's because it's gone into the cells themselves, which will cause swelling, and that means the cell isn't going to work as well. There's lethargy, headache, also possibly confusion, seizures, and coma. So this type of hypertonic hyponatremia is a slightly different reason to have the fluid shift we talked about, where we're pulling water from the cells into the extracellular space, where we're shrinking cells or moving water from the interstitial space into the vessel. This is an example, a different type of example. So in this situation, when we talked about the hypertonic water loss, there was cellular dehydration and dehydration in the vessels. In this instance, it was because water was lost in excess of sodium, so the concentration in the vessel increased because of the sodium. But sodium is not the only reason the concentration in the vessel can be different. The other factor, which has more pull than sodium does, is glucose. And if a person has profound hyperglycemia, they will have a hypertonic solution. Inside the vessel will be very, very concentrated because of the sugar. And that will pull water into the space, into the vessel space. So people's sodium is actually low because it's diluted. And if the kidneys are working, everything's working, they're going to urinate that all out. So the person with diabetes has polyuria because the fluid has moved into the vessel space and then leaves the body, is filtered out. So they will have profound dehydration, cellular shrinkage, but hyponatremia instead of hypernatremia. So go over that a couple times because it's easy to say it backwards. Having taught this particular section a couple of times, there's been times where I said it wrong the first time, which is very frustrating. So just be sure you understand which way fluid will move, and if it's, if the question is asking you between extracellular spaces or between intracellular and extracellular spaces, be very careful when you're reading that. All right, and one last thing to look at, which shouldn't take too long, not as fast as the cancer thing I did, but is just some more information about the electrolytes that we were kind of talking off and on along the way. Be sure I'm still sharing, yes. So we have our fluid compartments, our extracellular fluid and our intracellular fluid. And besides fluid in those compartments, there's concentration of particular ions. Positive ions, which we talked about already, intracellular and extracellular ions. Well, all these parts, they are actually. You might have looked at it before they were open, because I hide things, because I don't want people to look too far ahead. So if you go on our Moodle page now, the class from June 4th is there, and it has all the PowerPoints we talked about, some videos, as well as a copy of that very short case study we did. But that opened up like a day or so ago. So if you looked at it before, you might not have seen it. Um, and then I have recorded this, although I didn't start recording in the beginning, but I'll upload what I did record of this Skype session. So anyway, so here's our distribution of positive ions between the extracellular fluid and the intracellular fluid. And as we said before, there's more sodium in the extracellular fluid and more potassium in the intracellular fluid. And then this is the separation of the anions, or the negatively charged ions, more chloride and bicarb in the extracellular fluid, and more proteins and phosphorus in the intracellular fluid. So we have to maintain that balance because we need to keep the correct balance of charge across the cell, because we want the cell to have its resting potential, negative resting potential, and we want the ability to move the ions across the membrane when needed. And we want to be sure we keep the correct amounts inside and outside the cell, because as we talked about before, we don't want a lot of calcium inside the cell free. We'd rather have that magnesium there, and that's how we maintain that balance. So we got to be careful. So potassium is our major intracellular cation, big contributor to the action potential, cardiac muscle, and muscle contraction, because it's the potassium's largest role in the action potential is actually repolarization, and when we can allow the next stimulus to occur. So potassium is absorbed into the intestine, there's moved into the cells, so we don't have a very high potassium inside the blood vessel. Its concentration is maintained by the kidney, and any excess is easily excreted and filtered by the kidneys, as long as the kidneys are working. And so if there is a change in intracellular potassium, it certainly could be related to kidney dysfunction, because we want to be sure that the potassium is filtered correctly. It could be related also to a shift of potassium from the inside of the cell to the outside of the cell for reasons that aren't normal. It could be a change in the available hormones, an increase in insulin, and an increase in epinephrine will increase the movement of potassium. It could be a change in acid-base balance. Hydrogen is associated with an increase in acidosis, and one of the cell's buffer systems is to exchange potassium for hydrogen. And so in the setting of acidosis, the body will try to hide some of the hydrogen ions by exchanging potassium and hydrogen. So that can lead to hyperkalemia. Hypoxia can lead to hyperkalemia because hypoxia might lead to cellular necrosis, as we talked about. If it's that oxygen isn't corrected, then the cell swells and potentially lyses, opens, and releases potassium into the extracellular space, or direct injury to a cell that causes the cell to open, like a burn or something, could lead to an increase in potassium. Reasons why we might have a decrease in potassium could be diuretic use, because especially if it requires, if we use ones that aren't potassium sparing, could be loss through fluid loss, vomiting and diarrhea. These are reasons for low potassium, or the opposite of acidosis, alkalosis. So if the potassium is low, it's harder to repolarize. So it's harder to get back to the resting potential, because that is the job of potassium. So If the can't get the potassium level outside the way I'm supposed to because the level is low, then it's harder to get another action potential generated. I can't remember who it was that talked about hyperpolarization or the need to repolarize earlier in the class. So if that occurs, then things move slower. There's muscle relaxation, decreased shallow breathing. It could lead to cardiac dysrhythmias, but not as bad as hyperkalemia. That means that it's more likely to lead to a Brady dysrhythmia. If the potassium level is high, for the reasons that we talked about, the manifestations would be muscle irritability. Because the potassium is high, it means it's easier to repolarize the cell. And that means the cell can accept a new stimulus easier. So it's excitable. And the biggest concern, and the reason why hyperkalemia can be quite dangerous, is because it can lead to ventricular tachycardia or VFib. So that has to be corrected faster. Calcium, phosphorus, and magnesium. So we saw calcium is supposed to be outside the cell more than inside. Inside the cell, it's supposed to be packaged. Phosphorus and magnesium are inside the cell. These are supposed to sort of work together and move together. They're ingested, absorbed in the intestine, eliminated in the urine. These ions, there's a large percentage of them located in the bone. They're part of the hardening or the hard structure of the bone. The regulation of calcium, phosphorus is largely regulated by the parathyroid gland. So calcium and magnesium concentrations are linked together, and if either are low, that'll cause an increase in parathyroid stimulation or release. And phosphorus concentrations are inversely related to calcium and magnesium. So calcium and magnesium move together, because remember they're that pump together, and phosphorus is inversely related to calcium. We do not want phosphorus and calcium to be high together, or it can cause crystallization of tissue. So calcium is highly regulated in the body. And the way this occurs is through the parathyroid glands. So the parathyroid glands are 4 glands that are located on the thyroid gland, and they are constantly watching, are sensitive to the serum calcium concentration. And if the serum calcium concentration is is low, and most of the time when that occurs, the phosphorus is high because they work opposite, or they're opposite. A low serum calcium is the stimulus for the parathyroid glands to release parathyroid hormone. And parathyroid hormone has several functions in the body. These functions are meant to increase the amount of calcium circulating in the blood. So one of the first things it does is it increases bone breakdown to release the calcium and phosphorus that are present in the bone. So they're sort of our storage depot of calcium and phosphorus. So we release them together. So we know that we want calcium to be higher, but we don't want calcium and phosphorus to be high together, or our tissue will harden. So, but we had to release both because they live together in the bone. So the rest of the jobs of parathyroid hormone are to save calcium and get rid, and some of that gets rid of phosphorus so we can keep the phosphorus level low. So one of the actions of the parathyroid hormone, besides allowing calcium and phosphorus to be released, is to cause the kidney to reabsorb calcium that's in the urine back into the bloodstream, and to allow for the excretion of phosphorus in the urine. We can get rid of a lot of the phosphorus, but we'll keep the calcium that we got from the bones. Parathyroid hormone will help activate vitamin D. That is one of the, um, that is in the kidney. Its final activation of vitamin D, which allows for increased absorption of calcium and phosphorus in the GI tract. And so I will bring my calcium up, and because I'm allowing the kidneys to excrete excess phosphorus, I will keep my phosphorus level from getting too high so I can maintain those. So if I don't have enough calcium in my body and I don't have the ability to release the stored calcium from my bone or absorb calcium better in my intestine because I have a parathyroid hormone condition, or I am abnormally losing calcium in my blood, or calcium is binding to things it shouldn't, or it's sequestered in the soft tissue. If I have hypocalcemia, then I will have cardiac changes, bone fractures, because usually if I'm releasing lots of calcium from the bones, then they are more brittle. It'll change my eyes and hair and nails, and it increases neuromuscular excitability. And that is because one of the reasons is because um, calcium itself is part of the stabilization of the sodium channel. And so when I have not a lot of calcium, the sodium channels are open and a lot easier to start the action potential. And so people will have excitability, and that could mean cardiac rhythm changes. And you can check the calcium level with 2 physical exams. By tapping on someone's cheek will cause excitability, and tremor of the cheek muscle or increasing the blood pressure cuff will cause their arm to move. If I have hyperparathyroid activity or there's an increase in bone breakdown, there can be more calcium in my body, and that would have the opposite effect in terms of muscle activity. So those gates will be closed. It's harder to open them because there's more calcium, and so people have a decrease in neuromuscular excitability. Phosphorus and magnesium, usually if there is a loss, if there's a change in kidney function or intestinal loss, it usually occurs with low potassium and low calcium. And again, um, either one is going to affect the action potential. Phosphorus is usually associated, hyperphosphatemia is usually associated with hypocalcemia. But the main thing to understand about calcium is what is the stimulus to the parathyroid gland to increase serum calcium, and that's decrease serum calcium. How it does that? By breaking bones down and allowing for increased absorption of calcium in the intestine, increased reabsorption of calcium in the kidneys, and loss of phosphorus in the kidneys. So let's say, because oftentimes with hormone function that we'll talk about later, there is a negative feedback system involved. So in this instance, it would be an decrease in calcium causes an increase in parathyroid hormone, and an increase in calcium would be the negative feedback, would be the mechanism that would decrease parathyroid hormone. So if parathyroid hormone level is high and the calcium level is high, what does that tell you? I'll get to your question in a second, Amanda. Right, the negative feedback loop isn't working. So, because it should, if the calcium level is high, then that should be what is telling the parathyroid gland to stop producing the hormone. So that in general means there's a problem with the parathyroid gland, that's the issue. And usually it's an adenoma that for some reason there's an overgrowth of parathyroid gland and it's operating rogue. It's not listening to its normal negative feedback. So that would be the way to look at that. If the calcium level was really low and the parathyroid hormone level was really low, that means the parathyroid gland isn't working, and that's why the calcium isn't rebounding. There's a question here about the difference between serum calcium and ionized calcium. It's supposed to be, ionized calcium is supposed to be slightly more specific to the free levels of calcium because calcium itself can be bound to protein and so your serum calcium level is kind of an indirect measure of the free calcium. But if you really want to know the free calcium, especially if you have a reason to believe that there's less protein and so you're not sure how much to give weight to the serum calcium. So I'm not sure if I'm explaining that very well. So say I have this patient who's been sick for a really, really long time and their albumin level is really, really low. And their calcium level seems normal. But maybe it really isn't. Because maybe if I checked the ionized calcium, which is really looking at the free calcium, it'll be really high because there's no albumin for the calcium to bind to. So I'm trying to assess the actual free calcium better. So where I saw it used a lot was when I worked in the ICU because they don't know should the calcium actually be replaced or not? I wanted to know what triggered looking at. So probably they were unsure of how sensitive the serum calcium was. They were trying to decide, like, should I actually supplement this calcium? Because you want calcium to be normal, especially in a very ill patient, because you don't want them to have any cardiac irritability. But maybe what the serum calcium is telling you is wrong. It says low normal, but you check the free and it's actually high. And then if you would have supplemented, you would have caused the problem in the opposite direction. Yeah. So that's why sometimes I don't miss critical care. There was like 7,000 things to think of at the same exact time. Now, as a surgery NP, I can just look back and criticize them. I'm sure you do check it a lot in the CVICU. Now that I'm in surgery, I'll be like, oh my God, what are they doing? But I understand there's like so many moving pieces to that it's hard to keep all the plates up, as they say. So, oh my God, we got through it. That's a lot of material. I'm sure you all feel very stuffed. So that's why, I mean, there's so much we go through. It's helpful. I'm fine with people recording it on their own. I will try to remember to record it myself if that's helpful. That's why you do need to sit with the material and think about it and look at it again to be sure that you're not misinterpreting something or that you don't remember it correctly. In the next hour or so, I promised my kids I would play Uno with them. They're bothering me. So in the next hour or so, I will post the study guide for the first exam and the case study so that you have that. And then I will send a reminder so you remember that the next class, oh, I'm glad, Erica. The next class will be, the first thing will be that exam. Okay, Rebecca, do that. The next class will start with the exam and then did you hear the dog? He sort of whimpers sometimes. There's a chinchilla down here. This house is a zoo sometimes, and he really wants to meet the chinchilla, but the chinchilla does not want to meet him. And I'm in the basement in my office. So my dog is My dog's name is Sirius because I am insane about, oh, I can explain to you why that is. I'm insane about Harry Potter, so he's actually named for Sirius Black, one of my favorite Harry Potter characters from the books, because they're better than the movies, but I do like the movies. And I have two kids, 12 and 14, and we have done puzzles and we've played lots of games, and we have done kind of a lot of stuff since we've all been stuck at home together. So, Rebecca, if you the reason is this class is shared between two classes. I know, between two classes. So you are looking at MSN 621, which is obviously what this is, but all the course material is linked with a second class. See where it says the 625, 605, even though you're not on it. So go to your dashboard. There you go. Okay, so then go into 2020 CM. Not that you would have known that possibly, but and go in there. There's the class. You're in there too, obviously. Um, but that's where all of our stuff is. Sure. If you have any other questions, well, why would you? Any other questions, you can email me. And otherwise, I thought it was a fun first class too. We'll kind of get through this. We'll still get it all in. Sure, you're welcome. Nice to meet you guys. Thanks. Have a good night too, and I'll see you next week.
Good night.
Good night.
Bye.
Bye.
Good night.
Good night.
Thanks. Good night.